Hi everyone. I wanted to do an introduction to the Big Phylogeny. So we are going to start drawing that and we'll add to it for the rest of the semester. I haven't used the Zoom whiteboard yet, so forgive me if I need to make pauses or go back and fix things. See if I can move this out of the way. Okay, so the Big Phylogeny is big and it has a lot of stuff on it and it includes all of the groups that we've covered. So for now, we're going to leave plants as this big group here. Because we haven't covered any of those groups of plants yet. And so you can leave that space blank. You don't have to draw that big triangle at the end. But I am just drawing that big triangle because it's a huge group of organisms that sort of represents the end of our big phylogeny. But we haven't covered them yet, so we won't get any of those details in this right now. So two groups that we covered early on were the bacteria, and we did a little bit of archaea. So together, these are the prokaryotes. And you can imagine time as sort of going this way. Whereas as we get these points further up the line, we're sort of moving further in this evolutionary time period. And these points here represent speciation events, these common ancestors where offspring would have differed and led to different evolutionary pathways. So all of these organisms are made of cells and have DNA. So these are ancestral traits of our giant phylogeny of living things that we're looking at. So we could even separate out bacteria and archaea if we wanted to by putting a line here and saying that archaea have more eukaryotic-like DNA replication processes. So we do know that there's a trait that unites our bacteria, which is having peptidoglycan. So peptidoglycan in cell wall is a trait that's shared by all of our bacteria. So that's what this line represents here, is a shared trait by everything that is beyond that point, so our bacteria. So after that, all the other organisms that we're looking at are eukaryotes. So they have a nucleus. So everything beyond this point, unless I write loss of nucleus, we can assume has a nucleus. And because we've covered a lot of distantly related groups, I'm going to put some down here just to sort of illustrate how far they are away. Oh, I bet if I put it like that, what am I doing here? I'll get in the way of what I'm going to try to write. So we'll put it here. I'll actually have my branches come down. So here's animals. Closest related group to animals would be the fungi. And then our slime molds, the Myxogastria, are not in the same like larger part of the phylogeny. We could group animals and fungi together in the opisthecons, and then if we expand a little bit more, the Myxogastria are in the amoebozoa, and in the amoebozoa we would also find the dictyosteliomycetes. Dictyostelio. I did something wrong here. Mycetes. So that is the amoebozoa right here, and then this is the opisthecons. So here you could say, you know, one flagellum, and it's a whiplash flagellum. And all of these have glycogen as a storage carb. So this is a trait that unites this group. And fungi have chitin in their cell walls. And that could be a trait we could use to distinguish fungi from these other groups. So you can add traits on these lines to kind of further tell the story of how we're making this hypothesis about how these organisms are related. So we'll have another group of organisms branch off here. And these are going to be our heteroconts. So our heteroconts are the Oomycota, the Phaeophyta, and the Basilariophita, the diatoms. So all of these are heteroconts, and we could have a trait that unites them, the heterocont flagellum, or flagella, sorry. For those traits, we could also put cellulose in the cell walls, and maybe that would be lost in the Basilariophita. It doesn't unite all of them. We could also put something like diplontic life cycle, because when these do sexually reproduce, all of them have a diplontic life cycle. But heterocont flagella is how we really tell that these are all related. So the oomycetes are the heterotrophic group here, and the other ones become autotrophic after secondary endosymbiosis. So that secondary endosymbiosis and gaining of that four-membrane chloroplast unites our Basilariophita and Phaeophyta. Another trait we could put here is chlorophyll C, because that is a pigment that both of those share that wasn't present in the ancestral group. For diatoms, we can separate those out because they have a silica frustule. So we can just add traits onto here to support why we are making these hypotheses about how they're related. Okay, so after this, all of the organisms that we cover are the result of primary endosymbiosis. So that acquisition of that cyanobacterial ancestor that then became a chloroplast inside some pre-red alga. So that pre-red alga might have been something like a glaucophyte. And then we get Rhodophyta, our red algae. Sorry, this is all squished onto here. It is a giant phylogeny that is very difficult to fit on one document. So we have our glaucophytes and our Rhodophyta, and the thing that distinguishes them, remember the glaucophytes had that cyanel with the remnant peptidoglycan, which wasn't a true chloroplast. So we can put true chloroplasts here. And the red algae have a few traits that unite them as red algae, but differ from other groups. So loss of flagella. We could also put the Floridian starch. And we can add the alternation of generations with carposporophyte. All of those are shared by the rhodophytes and aren't present in the previous groups leading up to that. So these are all things that unite red algae together. So after the red algae, we get the green algae. And we have Chlorophyta as a pretty solid group, but there's a lot of change going on right now with how we are talking about the green algae. The research that we're using calls these the streptophyte clade. So these are the streptophyte algae. We can't call them the streptophyta, streptophyta, I think that's how you say it, because streptophytes actually include all of land plants. I am squishing everything together unnecessarily. I forgot I have this whole area where I'm not talking about plants yet. Okay, so we have green algae here. Algae. And so the streptophyte clade of algae is with land plants in this monophyletic group. So they all descend from the same common ancestor, and you can't really separate out certain groups of the green algae from land plants. So they're all together in this big group called the Viridiplantae, or green plants. So if we look at some differences here, we have differences in the pigments. So we get chlorophyll B and carotenoids. We lose the phycobilins. And we get starch stored in plastids. So there's other traits we could add there. Cell walls that contain only cellulose, so we don't have those extra compounds that we find in other algae. Many things we could put. So those traits are going to be shared by all of these future organisms, all the land plants. And then this big section that we're approaching is going to represent all of the changes that happen between these kind of freshwater algae that are moving onto land and our land plants. So they're going to all represent these big changes and big evolutionary adaptations toward movement on land. So what are those changes? A big one is going to be the evolution of sporopollenin. And in our first early land plants, they're going to reproduce with spores. So they'll have sporopollenin in their spore walls. Later, when pollen evolves, we get sporopollenin in the pollen walls. So that's why it got its name, sporopollenin. It's one of the toughest polymers out there. And so it is highly desiccation resistant and decay resistant, actually. So sporopollenin is going to allow these organisms to put their propagules out there into this dry, harsh environment and allow them to survive better. So we get sporopollenin. We also get embryos. So an embryo is going to refer to this multicellular structure that's retained by the female gametophyte. And so the female gametophyte is going to retain that embryo and feed it. And so that's called matrotrophy. Eating of the mother. So that matrotrophy and retention, sorry, I keep making these weird lines, of our growing sporophyte is going to give it increased survival potential. So we are kind of nourishing and protecting our next generation. So we get sporopollenin to protect us from drying out, and then we get embryo, so matrotrophy. We protect and feed our growing sporophyte. All plants are also going to have alternation of generations. Sorry, I ran out of room on the bottom, so I'm writing it up here. So alternation of generations, we have a multicellular haploid and a multicellular diploid phase. So we're alternating between the gametophyte generation and the sporophyte generation. And our gametophyte is going to retain and nourish that sporophyte. They're also going to be complex multicellular. So while we have unicellular green algae, multicellular, we have unicellular red algae, unicellular green algae. All of our plants are going to be multicellular, and they're complex because they have tissues and multicellular gametangia. So gamete producing structures. feel like I'm missing something. Oh, terrestrial. So all these land plants start out as terrestrial, right? So we're moving on to land. However, there are plants that evolved in this lineage that then move back into the water, much like mammals, right? So we move on to land as animals, and then with some of our groups like mammals, some of those mammals move back into the water. And that happens with plants. So plants start out as this terrestrial multicellular group, and some of those descendants will then move back into the water. Okay, we're going to stop here. Throughout the rest of the course, we will continue to add on to this phylogeny, and we'll create what's in this triangle.